Um, I'm really excited to be giving this talk. It's kind of different than what I'm usually doing. Usually I'm either giving here's the cool project I've been working on, or here's Mira, but this is a more general talk about segmentation in general, using deep learning for it. And so I'm kind of excited to give this. Are my slides going to advance? There we go. This is more to keep me on track than anything else. Like I said, since this is a new talk for me, I really want to start with giving a little bit of background on myself. I think that deep learning segmentation in general sounds like a very intimidating topic. And so I really want to tell you about how I have a super non-traditional background and anybody can do deep learning segmentation. I'll tell you about the project that really got me started down this path. And then I want to go into a lot of the softwares that are available to you to use, more generally how to get started doing segmentation, tips and tricks for training your data, things like that. And then I want to highlight a couple of projects, both that I've been involved in and some of the ones that I've found that are my favorite, of why we segment our data to begin with. Like what's the point, what's the use, all of those things. And then I'll end with giving as many helpful resources as I have found to hopefully get you started. Like I said, I had a super non-traditional career path so far. Even before what's up here, I was homeschooled for all of high school and my primary education. I went to undergrad for biology, and then I just started working. I was a veterinary oncology nurse for nearly 10 years before I went back to school. And so I have zero research background prior to starting my PhD. I am not a computational person at all. I went to do my PhD in biomedical science, and I was like, hey, I'm going to do cancer. I really love oncology. I'm super passionate about that. And then I met my advisor who started telling me about cryo-EM, and I was immediately sold. And I abandoned oncology immediately and went right down that path. And during my PhD is when I started sharing the segmentations that I was doing on Twitter, on social media, because my advisor, again, was, I would get bored and segment things, or I had collaborations that I was doing that I was sharing stuff, and I was just sharing it to my lab. And he was like, you have to share this to everybody else. And so I started posting on Twitter, and I got noticed. I got a lot of attention, and it was really fun to start building new connections and new collaborations with that. But it is also ultimately what got me my job at Thermo Fisher, because my boss at the time reached out to me and said, hey, I have this data set. Can you do it? And it was this gorgeous, it was volume EM actually. So it's cryo slice and view data that he gave me. And I'll show it to you at the end of this talk. And it was gorgeous data. And I had so much fun playing with it that 2 days later, I gave it back to him completely done. And at that point, we started talking about a job. And so I joined Thermo Fisher. I am a scientist on the application development team, which is a terrible name for our team because it describes exactly none of what we do. We are a team of cryo-EM specialists and volume EM specialists that really, our whole job is to collaborate with researchers and help push their researchers forward using our resources. So I get to do all segmentation all the time now, which is fantastic for me. This is really the tomogram that started it all for me. This is when I joined Matt's lab, we were researching neuron development. So we were looking at neuronal growth cones in developing neurons. These are highly dynamic structures at the tip of growing axons. They are the guidance machinery of your neurons that are developing. So they guide them, they use extracellular and intracellular cues to guide a developing axon to its synaptic target. So they're really fascinating, and they're actin rich. So they send out filopodia that are just bundled actin, and they're really fun to research. But when I first joined, Matt was showing me these data sets that he'd already collected. He says, I have about 150 data sets already, and we're seeing actin filaments in 2 different conformations. So we're seeing actin filaments here that look what he was calling at the time hyper twisted. So actin is a long filament that's helical, so it twists along itself. And we could easily see regular actin. So regular actin's over here. It's got a known helical pitch. It was very easy to identify. But the actin that you can see here in the red box is much wavier. It's really hyper twisted. Its helical pitch was much shorter compared to F actin. And we had no idea what it was doing. We didn't know. We were seeing it in very consistently in almost all of our data. And so his big idea when I joined the lab was, hey, let's find all of this hypertwisted actin in our tomograms and let's quantify it. Let's understand its distribution. Let's understand how much is there. And hey, by the way, the best way to segment tomograms right now is by hand. So that really, really quickly pushed me towards segmenting things using deep learning because I wanted to graduate eventually. And also during this time, COVID happened. And so my project went exclusively home with me online. And I started learning how to segment tomograms using deep learning from the perspective of somebody who is not a computational scientist, who is not a deep learning specialist. It was definitely an uphill climb, but now that I've done that, I really truly believe that anybody can get there because these are the results that I was able to end up with, which is that I was able to learn how to train a network that could really robustly identify both actin and what turns out to be cofilactin, which is cofilin declarated actin. And once we got to this point where I was able to say, you know, all right, I can segment these two filaments pretty accurately, pretty robustly. Well, why not just do all of the structures that we can see in all of our data? So what you can see here in the middle is a growth cone on an electron grid. So it's outlined in this pink dotted line. And over top of it is overlaid 26 different tomograms that we collected over this single growth cone. We're able to overlay those exactly where they were taken from the search images. And I was able to segment all of them with one network that I've trained to look for actin, which is green, cofilactin, which is red. Yellow you can see is the carbon edge, but then there's also cyan membranes. When you get further towards the center of this growth cone, you're seeing tons and tons of microtubules. That's the dark blue. And then we're also finding two globular proteins, ribosomes in orange, and a chaperone protein in pink. And we were really excited with this. You know, this is working really well. So now it's time for the whole data set. And so over the course of about 60 hours is all it took for doing this. Once the network was trained, we segmented 21 different neurons, 106 data sets over these 21 neurons. And we were able to use this to, you know, kind of quantify the distribution of actin, cofilactin, and all the other proteins that we were segmenting in these neurons by cryo-ET. And what ultimately we found with live cell imaging from my lab mate and, you know, the combination of these two is that these filaments are regulating the filopodial dynamics and structure in these really highly dynamic outgrowing growth cones. And so this was really by far the project that jump-started my, I don't want to say passion, it's an obsession with segmentation. I honestly segment for fun. It's ridiculous. I'm possibly the only person I've ever met who does this, but I love seeing cryo-ET data come to life in color and in 3D. And so I do this all of the time. So where should you start? I think that's really the thing that holds people up a lot is there's a lot of programs. It seems super intimidating. And so my hope with this over the next few slides is to kind of give a very general brief introduction to what is segmentation and how I do it. And then an overview of some of the softwares that are available, both open source and commercial, some out-of-the-box solutions that don't require you to train anything. And then also some tips and tricks for training that I've learned through a little bit too much trial and error. Hopefully you don't have to go through the same path I did. Segmentation, I think when you're used to the term is a really useful term, but I don't think it's a great description for what I'm doing, where I'm, you know, I am taking an image and I'm identifying everything that's in the image, and I'm doing it in a way that I can teach the computer to do it for me. And so in this case, if I say, all right, every pixel in this image belongs to either the background, the pizza, or the pepperoni, I'm performing a semantic segmentation. Every pixel is identified. Every pixel is assigned to either, you know, class and label are the terms that are used kind of interchangeably. And in general, when I'm talking about segmentation, this is what I'm talking about. This is what the vast majority of image segmentation for cryo-ET is. One of the other really useful types of segmentation that is out there that is more challenging to do than semantic segmentation is instance segmentation, where every instance of, say, the pepperoni in this image is assigned a unique label. So we have pepperoni 1, pepperoni 2, and this is extremely useful for downstream analysis, for further quantification, but it is a much more challenging task. And then, of course, you have the most challenging task is kind of newer to me, actually, is what's called panoptic segmentation, where it's a combination of the two. Every pixel in the image is assigned to a label, and every instance of everything is assigned a unique label. This is extremely useful, but really not a very easy thing to do in an automated way. Usually when I end up with a panoptic segmentation, it's because I've done a semantic segmentation first. I've pulled out the class that I want to instance segment, and then I've combined them both together. I've never found a way to do this in an automated way. There probably is a way, but in general, these are the three that are the most useful, the most frequently talked about with cryo-ET data. Luckily for us, hand segmentation is no longer the gold standard. UNets, convolutional neural networks, are really, really good at image analysis and image identification tasks. The holdup or the roadblock that was kind of there for a long time was that just a basic convolutional neural network, which is what this is showing, usually requires quite a lot of training data, which was just, if you're going to have to provide quite a lot of training data, you might as well just hand segment your data anyway. With the advent of the UNet, which was designed for medical image segmentation, all of a sudden you could provide really quite small amounts of data and still end up with a segmentation network that was really accurate and really robust. And so UNets have really just exploded the field of automated segmentation, thankfully. I am, again, not a computational or deep learning specialist. I'm not going to tell you all about what exactly all of the layers of a UNet or a convolutional neural network do. But at a basic level, what you can think about is that a convolutional neural network is learning features in images. So they're passing filters over your image as they perform the convolution and pooling tasks. And it's learning features, edges, curves, things like that. And it's learning which features belong to which feature. And then when it's trained and you apply it to an image, it can take all of that training information and assign with pretty high accuracy, depending on your training data, each pixel to a class or a label. Thankfully for all of us, the basic training workflow for training a UNet is, regardless of software, pretty much the same. The steps to get you from point A to point B might differ, but in general, you are taking your data, you are hand annotating a little bit of it, usually 3 to 5 slices for me, and then you hand that to your UNet, you let it train, and once it's trained, you can apply it to your data, and you end up with a pixel, per pixel segmentation. So each pixel in your image is identified as one of the labels that you've trained this network to identify, either background or one of your proteins. That's it. There's nothing crazy between softwares. Like I said, the steps to get from point A to point B are different, but in general, you need hand annotated data, you need your UNet, and you need some way to train your UNet, and you end up with your segmentation model. The softwares that you can use to do this are plentiful. It's kind of up to you with your comfort level where you start, what you have available to you at your institution. On the open source side of things, I have less experience with these, but I am familiar with the offerings from Nepari and Fiji. They both have really big plugin communities. So they both offer manual and deep learning segmentation plugins. I know Nepari has a plugin for, if anybody's seen the Segment Anything model from Meta, that does like instance segmentation for you in a really iterative way. I know Nepari has a plugin for that. Again, I had not used these very much. I had access to both Amira and Dragonfly in grad school, so those were the ones that I used, and I'll tell you about those in a second. But I do know that these open source options are really fantastic, and they do have some really robust communities built around them that are helpful and have guides and things like that. iMod as well has segmentation software within it, so it has some manual segmentation tools. And it does actually have its own custom convolutional neural network that you can train onto your data. It's not a UNet, but it's kind of similar to a UNet. And it works decently well. It was the first one that I started using actually with our data for that project. On the commercial side of things, unsurprisingly, first up is Thermo Fisher's Amira 3D. So this is kind of I want to say a jack of all trades software. This is a very powerful image analysis software that can do just about anything you can think of. The problem is just finding how to do it. There are tutorials. It can be hard to find. And I think all of these commercial softwares can be very large toolboxes that are a little bit hard to find things in, but it works really beautifully. And I've actually gotten to see over the past year, they've focused a whole lot on really improving the segmentation options and the segmentation workflows. So I've been able to generate some really beautiful segmentations from Amira that I couldn't have done, a couple of years ago, just because I think it was just too challenging to get started doing it. Like their technical capabilities have always been there, but now they're really focusing on user friendliness. And so that's been a lot of fun to see and help with. This is the software that I am most familiar with, I would say, because this is what I used during my PhD. This is from Comet Technologies. It's called Dragonfly 3D World. They are a deep learning based software. So most of their offerings involve deep learning in some way or are guided towards that. They do also have plentiful manual segmentation tools and frameworks built in for training your own convolutional neural networks. They have a lot of different unit offerings and things like that. Last up is the one I don't have experience with, but I do know can handle cryo-ET data. So Amaras from Oxford Instruments. Again, manual segmentation tools, you can train your own networks in it. It's primarily, all the uses I've seen for it are with light microscopy, but you can absolutely use it for volume EM and cryo-EM data. I've seen that done before. I just have no personal experience. And all of these softwares offer free trials, offer lots of tutorials and tools to help you get started. The out-of-the-box solutions that I mentioned are much scarcer. These are far more challenging to train. Our data is so variable. It is noisy. There's, ice thickness differences. There's alignment differences. Like accounting for all of that and training a network that it can really robustly apply to all of our data is just a monumental task. But MembraneSeg has done it very well. I've gotten to play with it quite a good bit now. And one of the things that excited me most about it, honestly, is that for being an open source software, it was easy to install. It did not require me to install, 25 different extra packages because, again, I'm not a computational person. So when you start asking me to make virtual environments and download all of these different implementations, I go blank. But MembraneSeg is a fully 3D unit. It segments membranes and cryo-ET data, and it does so with pretty high accuracy. So it's a really fantastic tool that if you're really interested in looking at membranes and just membranes, MembraneSeg is a really great out-of-the-box tool. It can also be retrained onto your data. So if it's not doing a fantastic job, they offer a workflow and a tutorial on how to train it yourself. So I really encourage you, if you're interested in segmenting membranes, try it out because it's one of the easier open source tools I've used. The other one that I have not used, but I'm familiar with because I've looked into using it and plan to soon is Depict, which was recently published. So that's a pair of units. So there's a 2D unit where they use for segmenting cellular compartments. So cytosol, nucleus. And they're also paired that with a 3D unit that can segment continuous structures like membrane or filaments, and also can do particle localization. So looking for ribosomes or whatever protein you can train it for. Their pre-trained networks can be applied to your data, and they work pretty well on data that it's naive to, so it hasn't been trained on it. Again, I have not personally gotten to use it yet, but I do plan to. But I do know that they also have a workflow for training it for your own data. So these are the two out-of-the-box solutions that I'm aware of. I would by no means state that these 3 lists of softwares are complete. There's probably many more. These are just the ones that I'm familiar with and would recommend as a starting point for anybody wanting to get into segmentation. So now that you've, spent however long finding whatever software you either have access to or the one that seems the least intimidating to use, what's next? Like I said, the basic workflow for pretty much all neural networks or for all UNets for segmentation is the same. You need training data. You need to manually annotate some data and provide that as training information for the network. How much, how good it needs to be, all of those things are obviously very important questions. How much is surprisingly little? I mean, as I have it written here, a convolutional neural network is only as good as its training data. It's never going to be better than the training data you give it. So it's really important to give good, accurate data, but you really don't have to give very much to a UNet to train it. In general, I start with 3 slices of data, and often it's 3, quarter slices of data. I try to get away with as little as I can just to see how good I can make a network without having to spend 10 hours hand segmenting something. So in general, 3 to 5 slices is more than enough for one network segmenting just either one data set or tomograms from the same data set. I'll get into larger data sets in a minute. But when you're doing your initial annotation for this training, the 2 things that I think are the most important are that your training data is accurate and it is representative. And what I mean for accurate is both of these segmentations as training data will result in a network that can segment membrane pretty well. There's no doubt about that. These are great. But the one on top is not nearly as accurate as the one on the bottom. If you look closely, you can see that I've painted, there's pixels painted on either side of this membrane. And so this is not going to be a network that is as accurately able to distinguish, say, membrane from microtubules or things like that, because I've given it, it's just kind of sloppy. It's not bad, but it's not as accurate as I think it should be to give good training data. And when I say representative, what I mean is that you need to make sure that your training data includes representations of all of the features that you want it to learn. All 3 of these images have microtubules in them, and all 3 of them look very different. So if you give it training data that only includes these 2 microtubules that are running, diagonally along here, it's not going to be able to find the microtubules that are here. So this is the upper or lower Z of a microtubule, and you can really only see the protofilaments here. But it doesn't look anything like this. And so you need to make sure that when you're providing training slices, that you have at least some representation of all of your data. And whether that is, if you want to be crazy and segment an entire microtubule, every single slice that contains the microtubule, that'll be great. That will be very representative. You don't have to. You can do a slice from the middle and a slice from the lower Z, and that should be enough to train it to find most microtubules. The caveat being, if you also have microtubules that are running into your image, you need to give it views like this, so that you can see this is what a microtubule looks like in cross section. Because these networks, unless you're training a fully 3D unit, these networks are seeing one slice of data at a time. And so if you only give it training data that looks like the microtubule on the top, it's not really going to find the microtubules on the bottom. So those are really my key points for training data, is that it has to be as accurate as you can make it, and it has to be representative of everything you want it to learn so that it can train accurately and give you good results. The question of what network to train is intimidating when you find out that there's just an endless lists of UNet. Like there's not just one UNet, there's probably 30 or 40 different implementations of UNets. They all have different uses, they all have different intentions. At some point somebody got, they came out with no new UNets because there are so many UNets at this point. I don't think that they've successfully stopped new UNets from being developed. But in general, for our data, just a basic UNet performs really well. You don't need to look into all of the more advanced implementations, things like that, unless you start to have more advanced segmentation tasks that you might need a more specialized network for. But starting out with a basic UNet is perfectly fine. That being said, there's one caveat to that, but this UNet that was trained for this data down below is a 2.5D UNet. So what I mean by that is that the dimension of your unit is important. So you've heard me probably already in this talk multiple times say 2D, 2.5D, or 3D. Let me explain what those are. So a 2D unit sees and segments a small portion of your data at a time. So it's seeing one slice of data and it's training on and segmenting a patch. The patch size of your network is something that you can define when training, and that defines how big that small portion is. A 2.5D unit is one that still sees and segments one slice of data at a time. But similarly to how we scroll up and down in data when we're looking at it to see if the signal that we're looking at is really there, these networks can actually see pixel intensities from multiple slices above and below. You can define how many slices above and below. And so they can make actually a much more accurate classification on the slice that they're segmenting. These are by far my favorite. And the patch size here, again, is the same as in a 2D. It defines just that small region. 3D units are different in that they're seeing and segmenting data in 3D. So they're looking at a full cube of data, and your patch size is defining how big that cube is. A 3D unit is, in my experience, by far the most accurate segmentation model. The downside of them is they take dramatically more data compared to a 2D or a 2.5D to train, and they are much more computationally expensive. The 2.5D network, when we've done experiments with this, really kind of starts to approach the accuracy of a 3D unit and is much cheaper to train and uses far less data. So that's why I typically start with a 2.5D unit. It's very rare that I feel like I'm just getting such bad results that I need to try a 3D unit. But when I say a basic unit, the dimension I do believe really is important. But other than that, other implementations of units are a more advanced topic that I think are not required for 99% of segmentation tasks. On the topic of patch size, that is something that you can specify when you're training a network, and it does make a difference when you're training a network. In general, more context, bigger patch size is better. But the trade-off there is that a larger patch size, again, is more computationally expensive to train, takes much longer, and you're ending up usually needing to provide more data because a smaller number of slices can't be broken up into as many patches with, say a 256 pixel patch size. In general, what I land on is 128 pixels. It tends to be able to get lots of context with most cellular features. It usually works fine. And I will say that pretty much exclusively when I am segmenting cryo-ET data, I'm using bend 4 data. So these are usually like 13 angstrom pixel sizes or 10 angstrom pixel sizes. I am not segmenting at 4K. I have tried it once, didn't work, took like 4 days, not doing it again. Segmenting at 4K is crazy, but possible. I really recommend segmenting at 1K data, so bend 4 times. This is one thing I do get asked periodically, so I thought I'd touch on it. If manual annotation is such a time sink and it's required for training data, what if we use synthetic training data? What if we fake our data and use that as training data? The idea behind that being is that you take a model volume built of whatever proteins you want, either with PDB models or with representative shapes. You simulate a tomogram from that. You're simulating, the types of noise that you're going to see. You're simulating the missing wedge. And from that, you can generate a pixel perfect training set without any manual annotation required because you have the original model that's split apart by intensity. So you can just say, here's my training data. I don't have to do anything. And in theory, it's an amazing idea. In practice, it's much more challenging. It's very hard to simulate what we're seeing in an electron microscope. There are 2 implementations that I know of that are doing this decently well. PaulNet is, I believe, a deep learning based implementation of this. Cryo-Tomosim actually came out of my PhD lab. is a MATLAB implementation. And both of these have been used to train segmentation networks. What I find them mostly useful for actually is that you can actually use it to kind of bolster your training set. So you provide real training data and synthetic training data, and it does improve the accuracy of the network to just give more context, more training data. I have not had tons of success training segmentation models that are trained exclusively on synthetic data. We've certainly tried, but I think it's just a matter of spending the time to do it. I don't think it's a limitation of this. I think eventually we will get to the point where we can simulate data well enough that we can use that to train a segmentation network and not have to manually annotate, except in, specific cases. So I do want to just touch on that this is one other topic with segmentation that's becoming a thing more and more nowadays. Also on the topic, denoising is, I think I kind of agree with the previous speaker in that it's very useful for visualization. I find it very useful for creating my training data. I will often denoise specifically so that I can create more accurate training data more easily, but I almost always train on the raw data because the segmentation networks don't really improve with denoised data. They actually do a really fantastic job on raw data, and that saves me a step. So if I'm trying to segment 106 tomograms, I don't have to denoise all 106 tomograms and then segment all. So for me, denoising really is only useful in the context of segmentation for creating that training data. I don't find it makes or breaks anything as far as accuracy for segmentation. And then finally, how, if I have a big data set, so say I have 106 tomograms that I want to segment, how do you optimize to do that? It's pretty easy to train one network for one data set and get a fantastic result. Getting one network to segment 106 tomograms or even multiple tomograms that might have come from different imaging sessions is a little bit more challenging. But really it goes back to this idea that your training data needs to be representative. And when you're looking for inference or generalizability, what you really want to find is data sets that kind of represent the span of what you're seeing. So this is from a collaboration that I was involved in when I first started with Thermo Fisher, and I'll tell you a little bit more about it in a little bit. But it ultimately ended up being, I needed to find data from 4 different tomograms to include in my training set in order to get really accurate results, a network that could understand what I wanted it to segment in all of the data sets that we were seeing. So when you're looking for a network that is inferable to multiple data sets, just look at your data set as a whole and take into account, are there differences in ice thickness if you're milling these cells? Are there differences in alignment, signal intensity, things like that? And if there are, make sure that you include multiple data sets in your training data so that your training is representative of what you want it to find. And I will say, I didn't make a slide on this, but I probably should have. But when you are doing something like this, it's really important, and this is something that I didn't know when I was first starting out. It's something I kind of learned by trial and error, is if you're using multiple data sets to train a network, you either need to combine all of them initially so that you're training all and everything at once, or if you were iteratively training a network, say I have trained it on one data set and now I want to add some more data from another data set. It's really important to include all of the original training data along with the new training. The reason that is it's really easy to bias a model towards a data set. It doesn't remember the initial training set it has. It's going to take the new training data and its previous information and say, all right, now I'm training to this. So if you give it all of the training data from all previous instances to the new one, you're going to end up with a model that is learning both data sets, not one that has learned this one and now is going to bias towards this one. So if you get to the point that you're trying to train a model that is going to be looking at multiple data sets, just make sure to include original training data at each iteration so that you don't inadvertently bias your model towards one data set or another. Back to the topic of instance segmentation, where you're taking each instance of an object and you're assigning it a unique label. Like I said before, this is a much more challenging problem. Units are not really, just a basic unit doesn't do this well. There are certainly implementations that could do instance segmentation, things like that, but the instance segmentation implementations that I have used are Amira 3D has filament and fiber tracing. One of their extras is for that. It uses cylinder correlation, so it's not deep learning at all. It's just using template matching and cylinder correlation to trace along filaments. But this pairs really beautifully with segmented filaments. So you can remove all of the background and segment just along your filaments, and then use Amira 3D for template matching and cylinder correlation. So if you have fibers that you want to instance segment, this is a beautiful way to do it, and it works very easily. TARDIS is another one that's more recent, that is a deep learning based implementation of instance segmentation for both. They can do filaments, I believe they can do vesicles and other structures as well, like different kinds of membrane structures. So I have not gotten to play with TARDIS, but from everything I've seen, it's a really exciting implementation, and it is open source. So the link there should get you to their GitHub. And then the last way that I know how to do instance segmentation, which is kind of the older school way to do it, actually, is just to do a watershed transform. It does not work on filaments at all. This is for globular proteins or things like that. But if you have, say, ribosomes that you want to separate, doing a watershed transform, the idea of it is that it is meant to mimic the water from a point rolling down. And so if you have multiple points and the water rolls down, and where it touches, those get separated as different particles. And so the way the watershed works in image segmentation is you have a single pixel in the center of each particle, and then you perform the watershed from that, and you end up with very beautifully segmented and separated particles. It's challenging to do in cryo-ET data. I've gotten it to work a few times, but it is an option if you're desperate to get your globular proteins separated. So those are mostly, the tips and tricks just to get started, the softwares, the things that I have. But I get asked a lot, why do you segment things? Like, what's the use of segmenting stuff? And I think, honestly, I get that asked more often with, non-experts in the field. I think people who are really used to handling cryo-ET data can think of just endless reasons to segment data. But my answers are always pretty much the same, that when we see more, we learn. Every time we've made advances in resolution and magnification and what we can see, we learn more about cellular structure. And just being able to see our data in 3D helps to conceptualize it so much more than seeing it as a grayscale volume. On to, analysis. Analysis requires localization. If I want to know how many ribosomes are in this image, I need to know where every ribosome is. So any sort of analysis on cryo-ET data as far as ultrastructure really does require segmentation, which, again, if you've done any work with cryo-ET, we know that, but when I get asked this question, that's what I answer. And then kind of more personally and more, I just like to sell segmentation to everybody. Honestly, this data is so engaging and so fascinating when it's displayed in 3D. I have so many conversations with people, both experts that are not in this field, non-scientists, just because I'm able to show gorgeous, engaging pictures. And so I think segmentation is a really fantastic tool for scientific outreach, because it really is a conversation starter for all walks of life. So the next couple of slides, I really want to kind of highlight these 3 points with a couple of projects of just things that new observations that we've found when looking through data, some really cool uses of segmented data for analysis, and then just a couple of really engaging tomograms that I've gotten to segment since starting with Thermo Fisher, actually. This first one is actually a project that has come out of my PhD project. So that big data set of 106 tomograms that I segmented, like I said, one of the proteins was a big chaperone protein. I will not use its full name because its full name is dumb. We call it TRIC. It is a big, about 1,000 kilodalton protein that is a chaperone. It's involved in protein folding of both tubulin and actin monomers, along with a large host of other proteins. But it's really easy to see in our data. And so I had trained the network just to segment it. I was segmenting everything I could readily see. But when we started looking through our tomograms after they were all segmented, what we found was that TRIC is in every single tomogram that we found it in, self-associating. So it was clustering together. It was forming long chains. We often saw it forming like little, we called them rosettes. So it was forming little rings of TRIC. And these aren't, we thought, okay, well, maybe it's associated with protein folding and, translation. Maybe these are near ribosomes, things like that. And they don't seem to be highly associated with ribosome clusters. So we have no idea what this is doing, but it's not described in the literature anywhere. It's something new that we're seeing. And it is a project that is, I like to joke about this in my PhD, that this is a project for another PhD student, but just a really cool observation that nobody's seen before and really only came out because we segmented this incidentally while we were looking at actin and other structures. One of the other really cool papers that came out recently they're using a combination of, fib milling, cryo-ET, single particle, and segmentation. They're studying the respirosome, the respiratory chain in mitochondria and chlamydomonas algae. And so like they've done a lot of in situ structural biology, but they're using segmentation to really map out the native landscape of native mitochondria and this respiratory chain. And so this is quite possibly one of my favorite segmentations I've ever seen. I just think it's gorgeous. It's a beautiful way to display the data, to display the research that they've done. But they have also used it for further analysis and for some of their single part, like their in situ work as well. And so I think it's a really cool project that highlights how you can combine all of these different techniques into a really useful study. And on that topic, using that same chlamydomonas data set, actually, so this is a collaboration that I got to take part in where I was able to use segmentation to help facilitate some in situ structural biology. So we're looking at, this was a really high throughput data set. So in total, I think we've collected 1,800 tomograms and uploaded those of chlamydomonas algae. And we're doing in situ structural biology on as many structural targets as we can find. And one of the ones that we really wanted to study was one of these membrane proteins. So this structure here in the corner of this tomogram is the chloroplast, and all of those membrane stacks in there are the thylakoid membrane. And this is a little algae organism. So this is the light harvesting complex of this organism. And all along the membrane, you can see studded proteins everywhere that are involved in the light harvesting. So there's, photosystem 1, photosystem 2, cytochrome B, tons of proteins. And they were really interested in looking at photosystem 2. It's huge. It's the one you can see most commonly along the exterior here. And what they found was that they were really having a really terrible time doing template matching. Most of our tomograms, we had denoised. This is a cryocare denoised tomogram, so it looks really gorgeous. But when you zoom in on it, what you find is that the membrane looks really, really gorgeous. But all of these proteins that are embedded in the membrane were kind of smeared out or blurry. And we think that that was really affecting the template matching. And the raw data is super noisy. So using just the raw data to do template matching wasn't working very well either. So this is kind of about when I started at Thermo Fisher, and I was like, well, maybe if we can denoise these enough, I can train a segmentation model to segment everything along the membrane that could potentially be photosystem 2, and then we'll go from there. And so that's what we did. We used a type of denoising called regression denoising to just try to clear up as much noise as we can so that I could train a network to segment everything studied along the lumen of these membranes. And that is where this data set comes from, or this movie comes from. In total, we segmented 41 tomograms and ended up with about 52,000 candidate proteins. You know, maybe these are photosystem 2. And so they extracted all of those, and after classification and refinement, none of which I had any hand in at all, because I don't do any of that. Sophie, my collaborator, ended up with, you know, a 19 angstrom structure, which is not crazy impressive, not super exciting by any means, but this is in situ. It's a really challenging target. And that was only 41 tomograms, and there's actually close to 300 in the data set that have thylakoid membrane in them. So we have a lot more opportunities to really improve this structure. And so this was just kind of a really exciting proof of principle for using segmentation for something that it's really not traditionally used for, and helping to further along some in situ structural biology. So that was a really fun project that I got to take part in, and hopefully we can improve the resolution of that as we keep iterating on the process. This is another project that I've found that I just find it really fascinating. So this is out of Danielle Grotjen's lab. So this is a pair of papers that came out that they've developed a workflow for both collecting data, but also segmenting, and then using those segmentations to really study mitochondrial ultrastructure. So membrane changes, thickness changes, things like that. And so they've developed it's a morphometrics pipeline, they've called it. And that was really cool all on its own. But then recently, very recently, this paper just came out where it's displaying, they're using it where they're looking at cytoplasmic ribosomes on mitochondria that are really, they're altering the local membrane. And so they were able to show with segmentation and with this morphometrics pipeline that there are, you know, measurable membrane differences from ribosome, you know, from protein import for the ribosomes that are bound to these mitochondria. And so it's really cool to see some of the novel ways that people are using segmentation to further their research, to answer some really cool questions that Cryo-ET allows us to ask. The last two are, our last couple of slides, I guess, are just a couple of my favorite tomograms that I have gotten to see from this chlamydomonas data set. And really, I think how segmentation has elevated them. So on the left here, you can see the mitochondria, and it's kind of hard to see exactly what's going on, but this mitochondria is undergoing fission, which was a really crazy thing that we managed to catch in this data set. I have never seen anything like this before. And so I got to segment this, and it becomes really apparent really quick that, you know, we're looking at a snapshot, we're not labeling anything. It's hard to tell if it's fission or fusion, who knows? But there's an ER contact site along the side of this mitochondria. And based on some papers, we're able to pretty confidently say, you know, this is probably mitochondrial fission because it's ER mediated. There's a very distinct ER contact site. And so it's really engaging to see stuff like this in 3D, but it's also a lot more easy, in my opinion, to analyze it and try to determine what's happening. This is quite possibly my favorite tomogram I've ever seen. I just think it's fascinating. So this is a mitochondria that is bound to microtubules, and so it's likely being trafficked somewhere in this chlamydomonas. It is in motion, it's going somewhere, and obviously when our snapshots you can't see that, but one thing that's really cool that came out when I was segmenting this is there's some proteins kind of between these microtubules and this mitochondria that we're not really sure what they are because they're filaments. They look to me like intermediate filaments, but I have no idea what exactly they are. But I got to segment this. And one of the benefits of working at Thermo Fisher is that I've gotten to work with a graphic designer who does some animation. And so we played around with this and we animated it. We have no idea if this is what it really looks like in movement. but like, how engaging is this? How cool to be able to show that the data we're looking at is in motion and we're just getting a snapshot of it. And so I can't get enough of seeing cryo-ET data in 3D. I just think it's endlessly fascinating, and it's been a lot of fun to work with now a graphic designer who makes my stuff even prettier by putting it in Blender and animating it. And so that's been a lot of fun. So I do want to end now. This is just a handful of useful resources that I've found and used or made. So Membrane Seg, like I said, has a really great installation guide and some tutorials on both using it and retraining it. One of my favorite open source resources to use because it was so easy. Napari also has extensive guides and tutorials. The image.sc forum is a really fantastic resource, especially if you're using an open source software. They have a ton of open source experts there that can answer almost any question you have. But you can also ask questions about commercial software, and you often will find people there that have experience and expertise in those. And so it's a really, really helpful image analysis resource. Both Amira and Dragonfly have video tutorials. And then there is the segmentation tutorial that I made in grad school. That's a step-by-step, both video and written protocol of segmenting cryo-ET data in Dragonfly. So I am sad that I don't currently have a YouTube to link you to of mine that has personal tutorials. That is a future project of mine. So maybe next year, but these are just some really useful resources that I've found to get you started. And that's pretty much it. So I'm going to end by showing you, this is the data set that I was given. It is like I said, it's a fib milled sample. So it's Hydra cryo slice and view. And it is of a full Chlamydomonas algae. So I got to play with this and segment this, and it's pretty safe to say, I think this is the data set that got me my job here. So I'll end with that and open it up for any questions.
Thank you, Jessica. Really nice talk. Do we have any questions?
Yes, no, great talk. I'm particularly interested in the mitochondria stuff. Can you go back to the is the mitochondria fission slide? That one, yeah, I'm just curious. Yeah, the other one. So in the segmentation, the yellow stuff, what is the yellow? Is it?
Sorry, Jessica, you're muted.
What?
She's muted.
Oh, she's muted.
Sorry about that. Let's see. Let me grab this again. So.
Yeah, mitochondria fission.
Yeah, so we have, I don't know what those are. I often don't know what I am segmenting. Which sounds terrible, but it's true. I'm often given data sets that I'm not given an identification of every protein.
Yeah, that's understood.
So we haven't really looked into exactly what proteins we're seeing, but what I did was just essentially took whatever I can see here.
Okay.
And I segmented that. So this is this part of this segmentation is done by hand.
Okay.
There was no need for me to train a network on it because this is the only tomogram we have of this, and it's like 10 slices. So I did that all by hand, but I don't know what I'm seeing there.
Okay. Can you like go back first for in that raw data? So I'm just very curious about that part.
Yeah, so I mean, like, there's very distinctly some sort of structure.
Yeah, there is something.
Yeah.
Interesting.
Yeah, it's really cool.
Outside the mitochondria, right?
Yes, so it seems to be on the outside, and like you can see already where the mitochondrial membrane is starting to close on either side, but it's not fully. It's a really cool tomogram that we don't really know what's going on. It's the only one. I mean, we have 1,800 tomograms. It's the only one in the data set that we've captured this.
Is this cryofiber milled lamina?
Yes.
It is. Okay.
All of these are milled on the Arctis, I believe. But yeah, everything is from milled lamellae.
Okay, what specimen, what cells?
Chlamydomonas algae. So it's
Oh.
all of this is chlamydomonas.
Okay, okay. Interesting, very, very interesting. It's not published yet, right? Or is it?
So the data set is publicly released, so it's EMD 11830.
Oh.
There's a preprint out about the data set as well. And it's in review right now. So
Okay, okay. Great. So I look forward to see the data itself.
Mhm. Yeah,
Okay, great.
absolutely.
Thank
Like, I mean, you can access the data right now if you ever
Oh,
want to.
okay.
I
Including the raw data and the segmented map, right?
The segmentations are not up yet. I'm still working on getting those uploaded to both EMDB and the Cryo-ET data portal from CZI.
Oh. Okay.
But soon.
Okay,
All
good.
of the raw data is there. The
Okay.
segmentations are very soon to come.
Okay, great. Another question. So you said that you collect 1,800 tomogram and then like a tiered series for this project. Oh, did you use like a special scan for data collection, like a how you collect data, because it's a huge data set, it can take a long time.
Yes, it was an enormous data set. So this is a very big collaborative effort. I think in total they collected close to 3,000 tomograms. So the 1,800 is all of the high quality ones that are published or released with the data set. Many overnight sessions, so with the milling, it was all done on the ArcGIS with the web UI. But I know the detailed methods. I was not involved in the data collection. That all of the data was collected before I even started at Thermo, actually. But I know that preprint has some pretty detailed methods about how they collected everything, because it really was a kind of a showcase of our data collection capabilities. So I think that's pretty highlighted in the preprint.
Where was the data collected?
Primarily in both in Brno and Eindhoven, I believe. So it was collected from Thermo Fisher nanoports.
Oh, I see. Okay, good to know. Thank you.
Hello, Jess. Really great talk. I have one question about the segmentation of filament. I'm wondering whether any of the software can segment filament as thin as 2 nanometer in diameter.
Are you asking if Amira can do it? Yes, honestly. So I've done some pretty crazy, oops, where'd it go? There we go. I've done some pretty crazy filament segmentations. It doesn't actually, it more matters the resolution of your data than the size of your filament. So I have done some volume EM segmentations of microtubules. So each microtubule was only like 1 or 2 pixels wide in the segmentation. And so if I can segment that, Amira 3D could do fantastic instance segmentation on that segmentation. So especially for either noisy data or very small filaments, my recommendation is to segment the filaments out of your data first, and then do filament tracing on the segmentation. But it should be able to handle very thin filaments pretty well. If you can see it in your data, you can segment it.
So you mean first do segmentation, then do the manual tracing?
Yeah, that's my typical. I don't often try to do filament tracing without segmenting first, because it's just a more challenging task. If I can segment my filaments and just give it a binary segmentation, so it's just a binary mask of here's filament and here's background, and then have it filament trace along that, it does a beautiful job, no matter how big or how small my filaments are.
Okay, thank you.
This is going to sound wild, but I don't know if anybody's familiar with the process of how tracing went back in the early, like to mid 2000s. But let me tell you, this is blowing. And I actually met Matt a couple months back. We discussed this at the Midwest Cryo Center. But yeah, I spent months doing this, years, actually. I spent 10 years doing this on serial section TEM,
Mhm.
on synapses, and I've lost track of how many I've done, but each set would be about 100 serial sections, usually closer to 300. And then each synapse would be anywhere from 10 to 15 images that had to be manually montaged in Photoshop. And then each surface, every membrane apposition had to be traced. So the
Deep
tools,
learning, deep
I
learning.
cannot tell you how wild it is to be like, oh, you can do 5. I'm like,
Mhm.
sorry, I just had to say, like, this is extraordinary from
It's
somebody
so
who'd
much fun.
spent so many years doing this the hard way. So just heads up.
No, I give you so much credit for doing it the hard way, because I looked at the hard way and I was like, no, I'm finding somewhere else to do it. I want to graduate someday. So
Well, see, I wasn't a student, I was staff. So it was,
Yeah.
this is what you're doing now.
So Michelle and Jess, what software did you use for the manual tracing?
I did Reconstruct. It was a tool set that came out of Kristen Harris's lab here at UT. It allowed for extremely discrete, like particle, like not particle, but you could create a class of objects and then have object 1 of that class, and then as many as you wanted, and it just, everything was a piece.
Yeah.
Because we were tracking, different types of synaptic vesicles, because you have like dense core versus clear, and so we wanted to track every object in the synapse. So, and it was not sophisticated, really. Like, you really could not build, like, it didn't have pipes tools like I think Fiji was developing at the time, but the the alignment tools in Fiji were really dodgy when you got into EM images. They were really more tooled for histological work at the time.
yeah, I mean, for me, it's my primary manual segmentations. It's almost exclusively either Dragonfly or Amira. And I wouldn't even go so far as to say it's because they're better than open source softwares. It's what I had access to. I think use what you have access to. And at this point, I think that there's so many different implementations and so many different tools that you can really do fantastic work regardless of the software. But nowadays I'm Amira and Dragonfly just because that's where I'm comfortable.